Thank you for the introduction and thank you to the organizers for having me back to give an update on this topic. I don't have any financial interests, conflicts of interest to disclose. And so here's a brief outline of my talk for today. I will start with kind of general principles and nomenclature for the field, give some examples on sources of pharmacogenetic variation, move into some examples in my area of work, which is type 2 diabetes. And then we'll close with reviewing some challenges and future opportunities in the field, as well as some resources for those of you who are interested in embarking on your own journeys in your disease of interest. And so I'll start by saying that adverse drug reactions are really a major problem. There's been systemic reviews and meta-analyses conducted. One study in over 280,000 individuals showed that over 6% of those had experienced at least one medication-related harm, and over a third of these were preventable related to side effects. And these adverse drug reactions are associated with prolonged length of stay, increased costs of these hospitalizations, and increased risk of death as well, especially in elderly populations where there is a lot of medications used for a number of conditions. And so this is really a major kind of healthcare problem. And these findings really underscore the importance of pharmacogenetics and potentially mitigating these adverse drug reactions and minimizing these healthcare costs and improving safety. And so pharmacogenetics is the study of how an individual's genetic makeup influences their response to medication. And this can be measured in terms of drug efficacy, looking at side effects as well. And so the goal is really to target the right medication for a given individual and really reduce medication exposure to for those who are likely to develop a complication or a side effect. There are a number of benefits to pharmacogenetics. One, as you might imagine, as I mentioned already, patient stratification. So again, targeting the right medication to the right person and avoiding exposure to unnecessary side effects. In pharmacogenetics, we can also do agnostic sort of genome-wide searches, which might also identify potential drug targets, and we can understand a little bit more about a drug's mechanism of action as a result. And it might result in kind of a novel drug development and new therapeutic avenues. And then finally, pharmacogenetics can allow for functional characterization. And so you can use a drug to kind of perturb an individual, and you can also measure whether that perturbation with a drug results in a physiological response that you can then measure and compare whether that response is different by genotype at a particular locus. And so when you do that, you might understand a little bit more about the mode of action of the gene product. And so it can give you a little bit more information if you have a genetic region or a variant that you sort of don't understand what it does. You can use a drug to sort of help you understand that as well. And I'll provide some examples later in this talk. So here's some key terminology. So we use the term pharmacogenetics as well as kind of the broader term pharmacogenomics, which refers to sort of genome-wide methods and approaches that kind of help decipher response to medications by genetics. We'll use these terms kind of interchangeably. It's important to understand the term pharmacokinetics, which refers to how the body processes a medication, and that can refer to absorption in the body, distribution throughout the body, metabolism by organs, and as well as excretion out of the body. And then there's also pharmacodynamics, which is how the drug acts on the body. And so here we're looking at kind of phenotypes such as like drug response and efficacy, as well as side effects. And I'll refer to these terms in the talk as well. In terms of nomenclature, when we think about genetic variation, we can obviously refer to that by, that specific RSID, but there's also the star kind of nomenclature, and this is in particular used when describing sort of drug metabolizing enzymes or transporters. And typically the star 1 allele refers to the normal activity of that enzyme or the wild type. And then subsequent kind of star numbers refer to having differences in that enzymatic activity. And so, for example, for CYP2C9, the star 2 allele results in decreased enzymatic activity of that cytochrome P450 enzyme. And then you can also use this nomenclature to describe genotypes. So homozygous carriers of the star 2 allele are described as star 2/star 2. We can also construct haplotypes, which is a set of linked genetic variants throughout a gene that may have functional effects. And so again, in an example I'll show later, you can construct a haplotype that contains multiple variants that all kind of contribute to decreased function of a given transporter. So not all medications may require kind of inclusion of pharmacogenetic testing. And so there are a couple of indications that come to mind. One is if a medication has a very narrow therapeutic index. And so this is, a medication where finding that right dose is extremely critical because beyond a certain window, you can have really devastating side effects. And so it's very important to be able to understand kind of the variability in dose and be able to kind of provide guidance on a dosing algorithm to avoid those side effects. We can also see sometimes that there may be differential response to a medication that seem to kind of correlate with ancestry, and that may reflect differences in allele frequency in a given pharmacogene or variant that is different by genetic ancestry. And so that also can lead to some sort of genotype guided approaches. Additionally, there can be high variability in pharmacokinetics or pharmacodynamics, which may incentivize you to sort of look for, the genetic underpinnings of that variability. And then pharmacogenetics could be considered when you observe adverse reactions for a given medication where the mechanisms are really unclear, and you might do sort of an agnostic search to kind of try and understand that as well. So in terms of kind of different approaches, obviously, the gold standard here is doing a randomized control trial. And once you have sort of the evidence to support the impact of a genetic variant, you would want to kind of study that in a fashion where you randomize individuals to having genetic guided therapy versus kind of the usual care and look at outcomes. But of course that, before doing that, you need to sort of have the evidence to be able to study this. And what has been done in many areas is the use of kind of pre-existing clinical trials that have collected samples to do genotyping and using those well-phenotyped studies to really help understand the impact of genetic variants in response to these drug outcomes that have already been measured in these RCTs. And so I'll also provide some examples of that as well. We can also use population cohort studies where you can examine a population exposed to medications and look at treatment differences based on genotype. Now many of us are using data that's available in biobanks, you can really leverage that and construct cohorts where you can study medication response, but there are obviously some challenges that come with that, which I'll talk about a little bit later. And then finally, you can sample patients based on whether they experience an adverse event or poor outcome and do sort of like a case control study and kind of look at genetics in that way as well. In terms of genotype selection for these studies, the earlier studies were really hypothesis-driven approaches, meaning we're selecting candidate variants, influencing differences in drug metabolizing enzymes or drug transporters, and then associating that with a response to medication. But more recently, the emergence of the use of polygenic scores has also allowed for hypothesis-driven approaches kind of based on the polygenic scores, where the idea is that the genetic underpinnings of a disease may have something to do with the response to medications for that disease. And so many people have looked at, risk scores for disease, and then we can also look at risk scores that are anchored on some pathophysiology or some pathway related to disease, and try to understand the response to medication from a kind of mechanistic perspective. We can also alternatively utilize hypothesis generating approaches. So these are, again, genome-wide searches using high throughput platforms where we're trying to do novel discovery for a drug response. And again, the availability of kind of genome-wide genotyping and sequencing the future sort of allows us to do that as well. In terms of phenotype selection, we can look at drug efficacy phenotypes. So this could be a change in biomarkers. So for example, in my area of type 2 diabetes, we can look at a change in hemoglobin A1C, which is the 3-month kind of average measurement of glucose control. And so we can look at the difference pre and post exposure to a drug and as our phenotype of interest. You can also look at relief of a symptom. And so in the psychiatry world, you might be administering or looking at survey outcomes for depression and other psychiatric diseases, and differences in that can also be a phenotype of interest. And then you can look at disease progression as well, so complications of having a disease, or even looking on the flip side as sort of disease remission as an outcome as well. On the flip side of drug efficacy, there's adverse drug reactions. So you can construct phenotypes looking at, did someone, yes or no, develop side effects. We can also measure lab tests indicating toxicity. So if a given drug causes, liver toxicity, you can look at sort of changes in liver function tests and things like that. And then these are more sort of pharmacodynamic phenotypes, but we can also look at pharmacokinetic parameters as well. So you can, for a given drug exposure, measure drug trough or peak levels, and look at differences by genetics with respect to that, kind of indicating maybe that there's something about a processing of a medication that results in differences among individuals. Okay, so I'll move on and then talk a little bit more about different sources of pharmacogenetic variation. And so the first example here is with respect to somatic mutations, which are genetic alterations that occur in non-germinalized cells after conception. And these types of mutations have really played a crucial role in cancer pharmacogenomics. And there's been a lot of success stories about sort of targeted therapies for oncology diseases. So one key example here is the BCR-ABL fusion gene in chronic myeloid leukemia. And so the vast majority of individuals with CML have this BCR-ABL kind of protein that's made as a result of a translocation that happens in chromosome 9 and 22. And this results in basically a constitutively active tyrosine kinase that results in increased sort of cell proliferation. And so what was developed was a drug called imatinib, or Gleevec as a brand name, which basically inhibits that pathway and prevents that excessive proliferation. And again, this has been very successful. I'm showing here the phase 1 study of the use of this medication in 6 individuals, where you can sort of see each line referring to a single patient's response. And they start out with very elevated white cell blood cell counts. And then you can see even within sort of the first 2 weeks that there's been a dramatic reduction in that white blood cell count as a result of this medication. So the success of this drug really exemplifies kind of the power of targeted therapy based on these somatic mutations, and has really demonstrated how they can really lead to effective kind of cancer therapeutics. Another source is with drug metabolizing enzymes. And so one key family here are the cytochrome P450 enzymes, which are mostly present in the liver, and they play a crucial role in drug metabolism, often converting these like lipophilic medications into more hydrophilic metabolites. And one key example here is with CYP2C19 and clopidogrel response. And so clopidogrel is a medication that is used for prevention of strokes and heart attacks. And it's an antiplatelet agent, but this medication kind of comes as like a prodrug. And so what it needs to do is to be metabolized. And one of the enzymes that does this is CYP2 C19. And so as you might imagine, if there are variants that result in reduced function, reduced activity of the CYP2C19 enzyme, then there's less production of the active metabolite, as you can sort of see in this pathway here. And then, there's less of this to be able to kind of provide kind of their antiplatelet effects. And so what was seen in this study was that they classified individuals based on their genotype into kind of how well they metabolize clopidogrel. And they found that carriers of a reduced function allele had lower levels, as shown in the top panel A, of the active metabolite. And then in panel B, decreased platelet inhibition in individuals who sort of were, these poor metabolizers. And then the question is, okay, well, we see this, but how does it translate to these like patient outcomes, right? And so what they found was that in those same carriers who had a reduced function allele and reduced platelet aggregation, that the carriers had a 1.5-fold increased risk of death from myocardial infarction or heart attack, stroke, or cardiovascular death. And then they also had an over 3-fold increased risk of stent thrombosis, which is basically like a clot forming in like a stent. So again, we're seeing that this is has an effect on outcomes. And so what is sort of the clinical implication of this? Well, the FDA did issue a black box warning for clopidogrel where they show that loss of function alleles can really reduce the efficacy of this antiplatelet agent. And in terms of kind of precision medicine, what has been done or what others have done is basically instead of using clopidogrel for people who have these reduced function variants is to then switch them to newer antiplatelet inhibitors where there isn't that kind of effect of that variant. Moving on, drug transporters are also a source of variation that can really influence drug pharmacokinetics. These are membrane proteins that affect efficacy and toxicity by affecting how sort of that drug gets transported and ends up affecting bioavailability, how the drug is distributed throughout the tissues, as well as excretion. And so an example here is with the SLCO1B1 gene, which encodes for a hepatic transporter in the liver, and then the association with statin-induced myopathy. So statin medications are cholesterol-lowering medications that are very commonly used, and they are handled by this transporter in the liver. And so as you might imagine, having genetic variants that reduce the function of this transporter can result in a decreased hepatic uptake of the statins, really causing increased systemic concentrations of the statins in the bloodstream. And then this can result in potentially increased risk of side effects. And so what we know is that clinically, statins are very commonly prescribed, but these sort of muscle-related side effects or myopathies can be quite frequent and can cause people to stop taking them. It can result in non-adherence, which has downstream consequences. And so what was done to kind of investigate statin-induced myopathy is a genome-wide association study that was conducted in 85 cases and 90 controls who all had been on 80 milligrams of simvastatin. And the cases had statin-induced myopathy, the controls did not. And what they found was this variant that was reached genome-wide significance for association with the development of myopathy. And it was in strong linkage disequilibrium with another variant, which I'll show in the next slide, which actually was the variant that kind of causes a decreased function of the variant. And so for those who carried it, what was found was that the odds ratio for myopathy was 4.5 per effect allele. So this is quite significant in terms of effect size. What's interesting about this was, again, the study was done in simvastatin. And so the question is, does this apply to all the medications in the statin class? And what was found was that this variant actually exhibits some statin-specific effects. And so the effect of the variant was the strongest in people who were on simvastatin and then different, and not really having an impact very much for pravastatin or rosuvastatin, which was shown in another study. And so what you can do is, do pharmacogenetic testing, look at people who might have this variant. If they do, you could make some adjustments, either transitioning them to a statin where the effect of the variant isn't really impacting their risk of myopathy, or you can adjust the statin dose and use something that's less, because there is sort of a dose effect with the development of these side effects. Okay. And then the last source of variation I'll talk about here is drug receptors and targets. And so these are genetic variation in the proteins that interact with these medications that are being used to kind of produce their therapeutic effect. And so one example is with respect to warfarin dosing. And so warfarin is a anticoagulant that's been used to prevent stroke, and it has a very narrow therapeutic range. As I mentioned earlier, that does mean that you have to kind of reach a very specific target to kind of prevent excessive kind of risk of bleeding. And as you can see in the figure here, there's a lot of inter-individual variability with what the daily warfarin dose must be to kind of achieve that therapeutic range. And again, that's sort of a reason why pharmacogenetics might be useful in this context. And so warfarin mainly exhibits its effect by inhibiting this protein called V-core C1. And what was discovered is that there are variants in the gene that encodes for this protein that can reduce the function of that protein, and that protein is crucial for kind of the vitamin K activation. And so people who carry these variants in V-core C1 have an increased sensitivity to warfarin doses, and may need actually lower doses in order to prevent kind of overshooting kind of that therapeutic range. And so to kind of demonstrate this, again, I'm showing a study that looked at the effect of V-core C1 haplotype combination on warfarin dose and the time to therapeutic INR. And INR is sort of the blood test that kind of tells you whether you're in that correct range for kind of blood thinning effect. And so there's a low dose haplotype that's illustrated as A and high dose as B. And what they found was that this haplotype was associated with warfarin dose, where the AA individuals had the lowest warfarin dose. And then they looked at outcomes like time to first INR in therapeutic range, and these AA individuals kind of got there the quickest, but they also were the ones who had the shortest time to the INR being greater than 4, which clearly would increase their risk for bleeding. And so based on these studies, there's been a number of warfarin kind of dosing algorithms that have been produced to kind of help guide the choice of the dose to kind of hit that narrow therapeutic window based on these haplotypes. But as sort of with a lot of genetic studies, as we know, many of these were developed in kind of European populations. And so what was found was that some of these dosing algorithms, while they performed really well in European populations, did not translate nearly as well when we looked at African ancestry individuals. And the reason for that is that the frequencies of these haplotypes differ by ancestry, and thus affect the performance of these algorithms, as kind of shown here in the map with kind of the differences across continental ancestries. And so this again really highlights that some of the limitations in our current studies of pharmacogenetics, where we need to really be able to study diverse populations and extend some of the findings so that they can be impactful for all populations. And then just kind of coming back to the clinical implication of this, again, sort of the updated labeling for warfarin was a result of these studies, and there have been multiple trials examining the impact of genotype-guided warfarin dosing. And as I mentioned, some of these had mixed results. Some of them showed improvement in outcomes and reduction in some of these adverse events, but some didn't really have an impact, and part of that might be related to the population that was being studied. And then one kind of key lesson learned from kind of the warfarin story is that much of this has actually become very not as relevant with the development of new anticoagulant therapies such as factor 10A inhibitors, which are medications that don't require this type of close INR monitoring, and they are much more straightforward in terms of dosing recommendations. So it's really made some of this work obsolete, but just illustrating that as new medications come on board, the study pharmacogenetics sort of has to keep up. Okay, and so now I'll turn a little bit to kind of applications in my own area of work in type 2 diabetes. And with that, I'll start talking about metformin, which is the most commonly prescribed medication for type 2 diabetes. But unfortunately, despite sort of the widespread use, it doesn't work for everybody. About a fifth of individuals fail metformin in the first 5 years of treatment. And in individuals with in youth with new type 2 diabetes, over half fail in first couple of years. And so this is really a problem. And to understand the sort of inter-individual variability of metformin, genome-wide association studies have been done with respect to glycemic response for metformin. So this is a study that was done a couple years ago where they uncovered that genetic variation in the glucose transporter gene SLC2A2 was associated with glycemic response to metformin. And the impact of this variant on metformin response was actually equivalent to about 550 milligrams of metformin, which is actually a reasonably clinical dose. And then also showing that the variant that influences response also had an association with a differential GLUT2 expression in the liver, so kind of providing some functional evidence as well. Our group has leveraged the availability of clinical trials to conduct a pharmacogenetics. And so, for example, we've looked at the Diabetes Prevention Program, which is a study looking at individuals who are at risk of developing diabetes, who were randomized to either intensive lifestyle modifications, metformin 850 milligrams twice a day, or placebo, and looked at the development of diabetes in this population. This study had collected samples that undergone genome-wide genotyping, and so then we could actually then perform a genome-wide association study looking at, what we looked at was a change in quantitative traits. As listed on the slide in response to metformin. And so we did look at the subgroups of the metformin placebo arm, looked at sort of gene by treatment interactions. So we published this genome-wide association study, and this is one of our top findings where we identified a variant that influenced our response to metformin. And in the study, carriers who had this variant had a 0.4% increase in 1-year change in hemoglobin A1C per copy of this deletion, which is consistent with a worse response to metformin. So here we can sort of see by the genotype that the change in A1C sort of increases with each copy. We also kind of looked at the allele frequencies in this diverse population, and that most of the effect was driven here by African ancestry individuals, again, highlighting that this is sort of a variant that might have an impact on this specific population. And so it was, I think, kind of good that, we've started doing these diverse studies where we can kind of uncover genetic variation that is that influences populations that are non-European. And then what we also saw here was that there was a gene by treatment interaction where the effect of the variant was only present when the metformin exposure was present and not really at all in the placebo arm that did not receive metformin. Another example of pharmacogenetics studies that our group has done is with respect to kind of drug perturbation studies. So this is Sugar MGH, which is a pharmacogenetics study of 1,000 individuals from diverse ancestries who have not had an exposure to diabetes medications. And so we gave these individuals drug challenges, at first with a sulfonylurea, which where the mechanism of action is to potentiate insulin secretion, and then followed by a couple days of metformin treatment, which kind of perturbs kind of hepatic glucose output as sort of the mechanism. And so this, again, briefly is a study where we, again, had DNA collection, gave people the single dose and sort of assessed kind of the physiologic response to the glipizide through these insulin glucose measurements. We also gave them a couple doses of metformin, following by oral glucose tolerance tests, in which we also measured kind of changes in glucose and insulin, as well as metabolomics, and also incretin measurements as well. And so this was a resource that was created for a number of reasons. One, we were able to conduct and publish a genome-wide association study on these acute drug responses, scopozet and metformin. But also it allowed us to assess the impact of known genetic variants for type 2 diabetes, kind of going back to the idea that understanding some of the variants that increase risk for disease may tell us a little bit also something about drug response. And so we had done this also separate analysis where we looked at a number of published type 2 diabetes variants, and then we looked at how these variants differed in terms of their response in our drug challenge and whether there were any differences with many of the markers that we measured. And what we found, for example, here is that this protective allele was associated with kind of increased levels of an incretin, which is a glucagon-like peptide. And there was high evidence of co-localization and sort of sheds a little bit of light as to the function of that variant, maybe has something to do with sort of incretin resistance. Here, I've been only talking about sort of metformin and earlier medications for type 2 diabetes, but as some of you may know, there are a lot of newer medications that have developed in the last few years that have a lot of cardiovascular, renal protective effects. And here I showed the treatment algorithm for type 2 diabetes, which can be very complex to look at. But genetics doesn't really factor into this. And so our goal here is to truly understand genetic determinants of response for some of these newer medications beyond metformin. And so our group is currently, again, leveraging the well-phenotyped data in clinical trials to kind of understand this. And so we have completed pharmacogenetic GWAS in the GRADE clinical trial, which is a multi-center randomized controlled trial of over 5,000 individuals who are randomized to one of 4 glucose-lowering medications on top of metformin. So here, as you can see, the sulfonylurea, DPP-4 inhibitor, GLP-1 receptor agonist, and basal insulin. And again, a multi-ancestry study with a mean follow-up of over 5 years. And the primary outcome of the GRADE study was to kind of look at time to failure of a medication, which was our outcome when we did our pharmacogenetic GWAS. And so this we're currently working on a publication, and so that should be coming out hopefully in the next couple of months. But again, another illustration of how we can really leverage that data available in clinical trials to kind of answer some of these questions. Okay. And then to kind of share some of my viewpoints on challenges and future opportunities in this field. One of the things I've been highlighting is that a lot of existing pharmacogenetic studies really unfortunately lack diversity. Here's a figure that shows sort of the distribution across sort of various populations of the studies that currently have been published. This was again, and as you're seeing here, lots of green on the page, illustrating that many of these studies were done in European populations and that the kind of proportion hasn't really changed a whole ton over time. So there's still a lot of work to be done here. As I mentioned a little bit earlier, there's a opportunity to utilize electronic health record-linked biobanks, which contain a wealth of data. Many of you probably are familiar with using the MPG biobank or UK Biobank, all of us research program. But this does require careful phenotyping. And so what needs to be done is you need to identify individuals with your disease of interest. Then you need to identify the specific medication exposure and ensure that there is a reliable exposure, whether that's through, so for some biobanks, there is availability sort of pharmacy data or claims data to kind of help you with that, but that's not present everywhere. But once you've identified a clear exposure of a drug, you also then need to have the availability of biomarkers or whatever phenotype that you're trying to measure pre and post medication, and that sort of needs to be anchored to your kind of exposure window. And then, of course, you need to have the genetic information as well in those people. And so as you sort of add on those layers of filtering, you can sort of envision that your sample size starts out with a very large biobank sample that kind of whittles down over time. And so as you apply these these filters. And so just, you know, a limitation, but definitely, you know, in working with consortia or pooling some of these studies and doing meta-analysis, you can still get to be, you know, in a reasonable sample size for genetic discovery. And then a couple of comments about sort of implementation on kind of beyond kind of the initial discovery. As I mentioned, there are always new therapies being developed that might overtake existing therapies. And so as you discover these medications, like with the warfarin story, some of your findings may not be as relevant with the development of new medications. We might find that some of these findings may have small effect sizes, and you really have to kind of convince yourself if this is a clinically meaningful result that you would want to take to the bedside. And then there might be kind of complex results that might be hard for practitioners and clinicians to interpret. And we obviously want to not exacerbate existing sort of disparities in healthcare. And so we want recommendations to really have applicability across all ancestry groups. There's also, you know, the issues with like availability and cost of testing. The cost over time has diminished greatly. So that's one benefit of that has happened over time. And then we also need to have sort of acceptance of doing this, you know, by patients and clinicians. We need to have protections in place for patients if they're going to have this data and have genotyping done. And so in terms of, you know, what's coming for the future, we, I think, still need to do a lot of work with obtaining kind of large prospective studies. There are a lot of clinical trials out there that could be leveraged, but some of them don't collect any DNA at all. And some of them are sort of, you know, done by the pharmaceutical industry. So it's sort of hard to have access to that data. We need to have a clear, strong evidence base to kind of support clinical implementation and need to have variants that are sort of well validated, not just like one finding that doesn't replicate anywhere, but something that you sort of reproduce and see over and over again, and that needs to be clear. In terms of integration of genetic information, we need to be able to kind of have the infrastructure available to kind of streamline testing and return of results and protection of confidentiality for individuals. And then we also need to train people to kind of have expertise in the field, whether that's the practitioners who might be providers who might be ordering tests or interpreting the results, and also doing the work to kind of establish that evidence base. Okay, and then here I'll end with some resources for those of you who might be interested. So the FDA actually has a couple years ago announced a collaborative review of the evidence to support some of these pharmacogenetic associations. So you can go on their website and they do have a list of sort of curated associations where there is strong data. And then there are a couple of sections where it's sort of like supportive data. And so you can kind of look there for kind of some of these well-established variants, some of which I've already mentioned in this talk. In terms of sort of guidelines for implementation, there's the Clinical Pharmacogenetics Implementation Consortium, and they put out these nice guidelines to kind of help provide some concrete dosing recommendations as an example for some of these variants that may be clinically actionable. So for the statin story that I shared earlier, this is a flowchart for individuals, like what to do for individuals who have a poor function variant in SLC01B1, where if you're prescribing you know, a high intensity statin, this is what you might do in terms of your actual statin choice as well as a dose. And so again, it provides some concrete guidelines for implementation. There's also GKB, which is a database where you can look up, you know, your favorite medication or variant or whatnot, and find published associations that are in the literature. And then they have some nice diagrams of sort of various curated pathways, some of which I used earlier in my slides. And so again, this is a nice resource that's been sort of curated for a number of diseases and medications related to them. And then if you know you really want to get involved, there is the Pharmacogenomics Global Research Network, where you know investigators can network with others in the field. They have these kind of meetings and present kind of research in progress, and they have grants to kind of support work along kind of any area of pharmacogenetics. So again, sort of an opportunity there for those of you who might be interested. And so just to close, pharmacogenetics continues to advance, but there's still kind of gaps in the field. There are opportunities for advancing precision medicine, as I mentioned, potentially identifying new drug targets, understanding underlying mechanism. There's still some challenges with respect to, complex diseases and kind of studying multi-ancestry populations. And we really do need to demonstrate sort of this impact on outcomes. And some of the earlier stories I told really have that, but others, you sort of have this association, but you really need to demonstrate some sort of difference in clinical outcomes. And then the future might, involve, as the cost of genotyping goes down, really doing preemptive testing. So for all clinically actionable variants, measuring those, and then having that available in the medical record, being able to sort of go to that information in advance of prescribing a medication, and then choosing your dose or agent based on that information, incorporating it into clinical decision making. And this is sort of already, started to happen. So for example, there's the VA Phaser program, which currently is offering pharmacogenetic testing for over 250,000 veterans at a number of VA sites. And then in Europe, last year, they published the impact of using a 12-gene panel that reduced the incidence of clinically relevant adverse drug reactions. So there is definitely a work that is being done in this area with some of these variants that have shown to have some effect. With that, I will end my presentation and happy to take any questions anyone may have. Thank you.
Well, thanks everyone for braving the rain to be here. Thank you for an
Yes.
excellent presentation. And we'll be back in about 10 minutes for the main session.
Great, thank you.